Okay, so for this deep dive, we're looking at something that, um, honestly, most scientists thought was completely impossible.
Utterly impossible. We're talking about sequencing ancient RNA.
Right, aRNA. From woolly mammoths, of all things, preserved in Siberian permafrost. And this isn't just, a few thousand years old.
No, we're going back a stunning 39,000 years
Hey.
and getting reliable, readable sequences. It shatters the old limits.
So let's break that down for you.
Yeah,
We all kind of know about ancient DNA. That's the organism's blueprint, right?
exactly. The big static instruction manual for building a mammoth. It tells you what it could do.
But the aRNA is totally different. It's not the blueprint.
No, it's more like the notes from the foreman on the job site.
Hmm.
It tells you what genes were actually switched on, what the cells were actively doing right before the animal died.
So we're talking about functional biology. In deep time.
That's the leap. It's a snapshot of life itself.
And the undisputed star of this particular study is a specimen named Yuka.
Yuka, yeah.
Mm.
A juvenile mammoth. And it gave them the most incredible, high-quality RNA from its muscle and skin tissue.
And the results were immediate.
Oh, yeah. Right away, they could see these were not just random degraded fragments. The RNA profiles they found in the muscle tissue were, well, they were exactly what you'd expect to find in muscle.
So things like titin, nebulin.
All the big ones. The core transcripts for building and maintaining sarcomere structure, for muscle function. It was like reading the cell's final to-do list.
But this is where it gets really fascinating. It's not just that it was muscle, but what kind of muscle?
This is so cool. By looking at the types of transcripts, they saw a molecular fingerprint for slow-twitch skeletal muscle fibers.
Which means what for a mammoth? Endurance.
Endurance. This animal was molecularly primed for long-distance travel, for navigating that harsh terrain, not for a quick sprint. We can actually infer its lifestyle from 40,000-year-old molecules.
That's amazing. But the aRNA didn't just add new information. It actually went back and corrected a really basic, a really old error.
Right, about Yuka itself. So, based on just looking at the body, the external examination, everyone had identified Yuka as a female.
For years, that was the consensus.
Yep. And then the RNA data comes in, and they found sequences that mapped directly to Y chromosome genes.
Wait, so?
So Yuka was XY, a male, the whole time. The molecular evidence just completely overruled what our eyes were telling us. It really highlights how powerful this is for ancient remains that might be ambiguous.
Which, I guess, leads to the big question of how. How did this incredibly fragile molecule survive for almost 40,000 years?
It seems the answer was the cell's own architecture. The RNA didn't just survive by chance, it was sort of packed in there, protected.
So what, the dense packing in the nucleus?
That's the theory. The tight chromatin structure, and maybe even the high number of mitochondria in the cells, it seems to have created this defensive vault, keeping the molecules readable for millennia.
So connecting this to the bigger picture, this really opens the door to what they're calling integrative paleo-studies.
Absolutely. We can now combine the blueprint from genomics, the structures from proteomics, and now the activity, the transcriptomics.
You can build a complete biological picture of an extinct animal, how it lived, how it responded to its changing environment. The detail is just unprecedented.
Which leaves us with a pretty provocative thought for you to consider.
If these dynamic active RNA profiles can survive for nearly 40,000 years, buried in permafrost.
What other biological insights are we missing? In the millions of other ancient remains we have sitting in museum collections right now.
Exactly. What other lost messages are just out there waiting for the right technology to finally let us read them?